Welcome, everyone, to this edition of the Della Pietra Lectures. The Della Pietra's goal in helping provide for these lectures was to bring scientists to campus and to the Simon Center, scientists who are doing innovative, exciting, important, influential work, and scientists who can explain what they're doing and the more general context in which they're doing it to a public audience such as this one. The belief is that this country will be much better off if the understanding of science is much broader than it is today, and this is a small attempt to move in that direction. And we're very pleased today to have an exemplar of this kind of scientist to speak to us. This is Stephen Quake from Stanford University. He received his BS in physics and MS in mathematics from Stanford University and his PhD from Oxford University, where he was a Marshall Scholar. After a stint at Caltech, at least partially in the lab of Stephen Chu, Nobel Prize-winning physicist, he started the Department of Bioengineering at Stanford, where he is the Lee Otterson Professor of Bioengineering and Physics. He's a member of the National Academy of Sciences, the National Academy of Engineering, and the Institute of Medicine. He has won many international prizes, including the Sackler Prize in Biophysics. He's known for his pioneering new approaches to biological measurement, explicitly to microfluidics. He made the first single molecule DNA, and he's one of the few people in the world to have seen his own genome. I'm not sure how he reacted to that, but maybe he'll tell us. Anyway, please join me in welcoming Professor Stephen Quake.
Well, thanks so much for having me here. It's a real pleasure to get to know the folks here. And they told me this was a general audience, but let me just check that here. A show of hands. How many people have a degree in physics or math in the audience? All right. Now, you must have the highest density of mathematicians here in Stony Brook in the entire world. That's about half the audience. Okay, that's helpful. Another show of hands. How many people know what DNA is? That's pretty good. All right. I'll tell you a bit more about it, and I'll give you a refresher too. So I titled the talk Precision Measurement Biology because I was brought up as a physicist, and in particular in a precision measurement group, and it's been such a powerful influence on physics over the centuries that, and it's just now beginning to make its inroads as a concept in biology, and so I like to use that as kind of a jumping off point. And so, in physics, it all goes back to Galileo 300 years ago. And he's sort of recognized as the father of precision measurement. There's this lovely quote associated with him to measure what is measurable and make measurable what is not so. Unfortunately, it appears to be completely apocryphal. There's no sort of scholarly evidence he actually said it, but if he didn't say it, he should have. And of course, what he did do was build the most precise telescope of his time, turn it up to the heavens, make numerous astronomical discoveries. But he was also involved in many other areas of science, including mechanics, and he was interested in sort of measuring the behavior of rolling balls and cylinders, and kind of had a forerunner to Newton's laws. He'd figured out the displacement was the square of time, and he measured those things very carefully, those curves. And he's, of course, such an important figure in the history of physics that his finger is preserved as a relic. You can go see it in the Science Museum in Florence, Italy. I was there this summer. It's really quite spectacular. And if you're ever there, I encourage you to drop in and check it out, along with many of his instruments and books. And so, I'll give you maybe another example of the importance of precision measurement in physics just from the 20th century. The sort of continued spectroscopy, focus on spectroscopy of the hydrogen atom led time and time again to sort of unexpected discoveries that have forced us to revise our theories of the physical universe, and also to many technologies that are now part of everyday lives. And Rabi, of course, who invented nuclear magnetic resonance as part of his attempts to measure the magnetic moment of the proton. That led, of course, to MRI imaging, to global positioning system, atomic clocks, and there have been many technologies that have come out of people's efforts to measure physical phenomena to higher and higher precision. And that story gets repeated over and over again in physics. And we're now at a point in biology where we're starting to see development of very powerful measurement technologies that are changing the field and allowing one to not only investigate very fundamental questions about biology, but are enabling sort of practical applications in human health and medicine. And the two that I've been involved with most intimately over my career have been the development of high-throughput DNA sequencing techniques and the development of microfluidic tools, which are kind of the biological equivalent of the integrated circuit or computer chip. And so I'm going to give you some vignettes over the course of the talk of how we're using these measurement technologies in different areas, and give you sort of a picture of how biology is turning from a sort of descriptive science into really an information science and hopefully fully quantitative science in the not too distant future. So this is a cartoon of a DNA molecule, sort of its very famous structure of a double helix. And so it's sort of like a spiral staircase, or ladder that's been twisted around, rather. And it's steel cable backbones here, and holding the cables together are the bases, which encode all the chemical information in the molecule. And so there's 4 letters in the alphabet, A, C, T, and G, which refer to 4 chemicals. And they're strung together in such a way that the cell can read them out and use them as templates for making all the proteins and structures involved in the cell and the organism. And it's been sort of one of the triumphs of modern biology that one can actually read all that out now, and not just in little bits, in dribs and drabs. One's been able to read it out in dribs and drabs for about 3 decades, but only in the last decade could one really read it out in out of a fire hose, enormous quantities of the data that's stored in DNA. And that's created new challenges, like how do you interpret it, but also provide a number of opportunities. So let me give you, before we dive into that, just a few numbers to give you a sense of scale for DNA and the information and code in your body. Each of your cells has about 2 meters of DNA. So if you took the chromosome from one of your cells and unraveled them, laid them out end to end, it would stretch out to be about 2 meters long. Not that long. Now you have about 75 trillion cells in your body. So if you took all the DNA in your body and laid it all end to end, it would go from the Earth to the sun and back a couple hundred times. That's a lot of stuff in your body right now. And how do you fit all that there? Well, the answer is DNA is a piece of string, but it's a very skinny, skinny string. It's 2 meters long. It's only a few atoms wide. And that DNA is wound up on little spools and sort of all compressed inside the cell. And so that's kind of amazing when you think about it. It's quite a phenomenal molecule. And so I mentioned that we're kind of reading it out now as if it's come out of a fire hose, and this is a figure that gives you a sense of that. It's from Nature magazine's 10th anniversary of the Human Genome issue, and this is years on this axis from 2000 2010. This is the cost to sequence 1 million bases of DNA. It's been declining exponentially, in fact, faster than the famous Moore's Law. So Moore's Law describes how computers get faster and cheaper by a factor of 2 every 18 months. This has been even faster than that. So it's really been sort of this enormous sort of seeding technological revolution where there have been lots of new creative methods and a lot of competition to bring them to market. So their axis is the amount of information that's been generated. And they're kind of calculating it in numbers of genomes here by counting the people, and this is, I think, billions of base pairs on this axis. And so it's been phenomenal and quite a ride. And you've probably seen articles in the popular press about this or that genome being sequenced, cancer genome, different organisms and whatnot. It's been great to be a part of. But there's also more skeptical views. This is a quote from an article by Nick Wade in the New York Times a couple of years ago, where on this sort of 10th anniversary, he pointed out that hey, what's the genome done for me? When you guys wrote a proposal to sequence the human genome and suck up $3 billion of taxpayer money, you promised me you're going to cure cancer, you're going to cure Alzheimer's, and a bunch of other common diseases, and it hasn't happened yet. So have I been ripped off? It was a very sort of vexing article for us in the field, in that while it's true that common diseases have resisted sort of solution through genomic techniques and otherwise, there have been many other benefits that have accrued from having the human genome sequenced, many of them sort of directly clinical in nature, that were impossible to predict when people were proposing to do the project. And like many things in science, it's very hard to make predictions, especially when they're about the future. And so that's Yogi Berra there, right? All right. A few of you got it. And so to sort of say literally that you didn't do everything you said you would in the proposal is, I think, a little beyond the pale. And one can sort of really estimate what the actual benefits have been from the genome. People have done, economists have made numerical estimates of value to the economy, value to medicine, and they're substantial. And tonight I'm going to give you one or two examples just from my own research in things that would have been very hard to predict when the Human Genome Project started. People weren't even thinking about personal genomes then, and now it's become sort of a routine reality. I'm not going to talk about that so much tonight. We can get into that questions if you want, but I'll give you a few other examples. The first one's a very clinical example, and it has to do with prenatal diagnostics. So humans are diploid, which means we're supposed to have 2 copies of each chromosome. We get 1 copy from our mom, 1 from our dad. And you can look at them under a microscope. They're big enough. So remember, if you took all these DNA molecules and stretched them out, that'd be 2 meters long of a molecule. But here they're all condensed, so they're wrapped around spools and wrapped up. And so not all the full 2 meters. And there's 2 copies of chromosome 1, 2 copies of chromosome 2, 3, and so forth. This particular individual, however, has 3 copies of chromosome 21. Which is not a good thing. When that happens, that's called aneuploidy or trisomy if it's 3 copies. And when you have 3 copies of chromosome 21, it causes a genetic disease known as Down syndrome, which you may have heard of. It's the most common genetic disorder for live births. And these sorts of aneuploidies, or mistaken chromosome numbers, affect close to 1% of live births. It's a pretty large number. And so doctors recommend that an ever larger fraction of expecting mothers should get some sort of diagnostic to test the genetic health of their fetus. And the way that's been done for years and years and years has been to take a big needle and stick it right in the mom's belly, right next to the fetus, and try and grab a few cells and take them out and culture them and make that karyotype that I showed you on the previous slide. And the sort of most common one is called amniocentesis, and there's another one that's often done now called chorionic villus sampling, but both the same kind of idea. And as you can imagine, this causes risk, both health risks, both for the mom and the baby. And in fact, there's fatalities from this test that are pretty substantial, close to 1%, maybe 0.5% if you believe the doctors who are sort of self-reporting on this. So certainly no less than 0.5% of times that this test is done, you lose the baby. And I got very much interested in this problem when I became a parent and saw this being done to my wife. First kid, this done for our second kid, and there's got to be a better way. And so I got interested in trying to find non-invasive ways to make genetic measurements. And it kind of stuck in the back of my head for a long time. And then one day I stumbled across there's a very old literature dating back to the 1940s, which sort of described the fact that there's DNA in your blood that's not part of any cells anymore. So you think about DNA as the blueprint for the organism. It occupies this very privileged place in the nucleus of the cell, where it's the template. Machines come in and copy the instructions, take them out to the rest of the cell. Proteins are made and so forth. But when cells die, they sort of break open, spill their guts into the blood, and the DNA from the nucleus gets thrown out there as well. It gets chewed up into little pieces and it just circulates in the blood as naked DNA molecules. And so if you take someone's blood, you separate all the cells out and look at what's left, there's DNA there in what's left. And these people observed that for the first time. It's kind of amazing. They were thinking about DNA as just a chemical analyte. This is before Oswald Avery had figured out the molecule of inheritance. This is their data from the paper. And they looked at people with a bunch of afflictions, one of which is pregnancy. And they noticed in the case of pregnancy, there were higher values of DNA and RNA in the blood. So they realized something was going on. And it turns out that was because some of the DNA in the mom's blood is from the baby. And so it turns out that, at the end of the day, early in pregnancy, it's a few percent of the DNA in the blood by mass is from the baby, and so most of it's from the mom. But there's a part from the baby, and it goes up over the course of the pregnancy. Can be much, much higher towards the end of pregnancy, a huge fraction actually. And so with that notion in hand, I began to think about ways to measure whether the baby had the right number of chromosomes. And stumbled across this counting principle, where if you count the molecules of DNA from a given chromosome, if you could do that, and you could do that precisely, you'd be able to detect overrepresentation of one chromosome with respect to another. And so we ended up doing that in the lab, and by we, I mean my student, Christina Fan here, who along with our clinical collaborator here, Blumenfeld, was able to do kind of a real clinical study at Stanford. So what she did was to take the blood that Yair and his colleagues had collected from pregnant women. She purified the cell-free DNA, sequenced it on these new sequencers, and sequenced about 5 million different molecules from the sample, and then took each of those sequences and mapped them back to the human genome. So we're using the fruits of the Human Genome Project here by taking the average human genome and using that as a reference and trying to figure out which chromosome each of these sequences sort of originated from. And they each vote for a chromosome, and then we go look for voter fraud or looking for a chromosome that's overrepresented. Since only a few percent of the DNA in the blood is from the baby, you're not looking for the difference between 3 copies and 2 copies. You're looking for the difference between 102 copies and 100 copies, right? Just a very small fractional change. So it's, in general, very hard to measure small differences in biology, and these sequencers provide us an exquisite way to do that. And so here's the data from our first little study. This is the relative representation of chromosome 21 in the blood of these moms with respect to chromosome 1, which is our reference. And you can see that all these red data points here, which are sort of elevated, are from women who are carrying fetuses with Down syndrome. And all the blue data points are from women whose babies do not have Down syndrome, and they completely separate. You can draw a line, and it's really quite evident. There's now, of course, nothing particular about chromosome 21 in these measurements. We're randomly sequencing DNA, getting it from all the chromosomes, and so we can look for trisomy or aneuploidy of any of the chromosomes. So we did, and this is now all the chromosomes here. They're in a funny order, which is not important for the talk here, but this is again the relative representation. This is chromosome 21. This is the ones I showed you before. Now, it turns out we had 2 women in the study who were carrying fetuses with trisomy 18, and it's those 2 outliers right there. We had 1 woman in the study carrying a fetus with trisomy 13, and that outlier right there. And so this showed you could identify trisomy of any of the major occurring chromosomes. So 21, 18, 13, those are the ones that are survivable and so the most important ones to test for, but you can of course do it for anything. So we wrote this up, published it in 2008. It got people really excited, set off an enormous amount of interest in the field, and a bunch of clinical trials were launched both in academia and in industry. They read out in about 3 years. And so, they collectively tested thousands of women, and it all worked just the way we said it would. So the test became available in the clinic. There's about 4 or 5 different companies that are offering it worldwide now. And this year, in 2013, something like 500,000 women have received the test, or will receive the test by the end of the year. And that's just, an enormous number. I mean, it's moved from sort of a little research project in the lab to something in the clinic much more quickly than I ever would have expected. You can do the math and work out if all these women were going to get amnio and did this test instead, it's a large number of lives saved, a couple thousand. Also, I was involved with one of the companies that did do the test. It was acquired by Illumina earlier this year, so I'm now essentially unconflicted. But it's moved amazingly fast. While the clinical tests were going on, the trials, I wasn't involved in any of that personally. We were back in the lab thinking about other ways to use this counting principle, look for overrepresentation for other things in prenatal diagnostics. And one of my colleagues was always pushing me, asking me, when are you going to be able to sequence the whole genome, never mind these aneuploidies? So we began to think about ways to do that and realize that this counting principle could be used not just at the level of the whole chromosome, but at segments of chromosomes and also right down to the individual base pair. And so Wei Gu, who was a PhD M.D. student in the group, was one of the leaders in this effort to measure the whole fetal genome non-invasively, which we succeeded in doing and published in Nature last year. And I think I'm not going to dwell on that, but if you're interested, the paper's available on my webpage. So after we published the first paper, word had gotten around Stanford that I was interested in non-invasive diagnostics, and one day I got a call from a cardiologist named Hannah Valentine who said, we're treating all these heart transplant patients to people who've gotten a new heart, and in order to tell if the immune system is rejecting the heart, we go and physically biopsy the heart every couple of months. These people go through the trouble of getting a whole new heart installed in their body, which is very painful, and they have to give up a piece of it. Gets ripped out every couple of months to see if it's working, and that's not a good situation because it's very painful for the patients, it's expensive, and not a terribly informative assay, anyways. And so we began to think about whether we could use cell-free DNA now in a different context to test the health of transplanted organs. And the key to this was to think about the heart transplant as a genome transplant. And let me give you a little cartoon that illustrates that. So imagine somebody before a transplant, any of you in the audience. Right now, all the DNA in your blood comes from your genome. Cells from your body, they've all got more or less the same genome. I'll talk about some exceptions later. But when those cells die, they go in the blood. And let's say your particular genome, all the polymorphisms that make that genome you, we'll call those red molecules. And so your blood is full of red molecules from your red genome. Now, you go get a heart transplant. And so someone else's heart gets put in your body. Every cell in that heart has someone else's genome in it, has the heart donor's genome in it, right? Let's call that genome green. So now, when cells from the heart die and dump their DNA into the blood, you get green molecules going around. So your blood would have, after the operation, after the transplant, you've got a mixture of red and green molecules circulating in your blood. Now, if your immune system starts to attack the heart and kill cells, that's what the rejection is, then you expect more green molecules to enter the blood, and the relative amount of green versus red should change, should increase. And that's the principle we use to investigate this problem. So we sequence the cell-free DNA again, and now instead of mapping it back to the average reference genome and just asking which chromosome did it come from, we ask what were the polymorphisms in these DNA molecules that would uniquely separate the donor from the recipient. And we were able to analyze the donor-recipient genomes ahead of time, so we knew where they were different, and we looked for DNA molecules that would fall into one bucket or the other. And this shows some of that data now. These are 5 different patients. This is the x-axis is time after the transplant in months. This y-axis is the amount of donor DNA or foreign DNA floating around in the blood as measured by the sequencing, and the blue curves are the ones to look at. At every single time point, these individuals were physically biopsied, and the pathologist looked at the biopsy to say whether they're rejecting or not. And in the cases where the pathologist said there was a rejection, we've highlighted them with red, the data points with red arrows. And you can see that very clearly the foreign DNA is very highly correlated with the rejection events as called by the pathologist. And in fact, even before the pathologists are calling rejection, a couple months before, you're seeing the cell-free DNA increase. And so it's actually an earlier diagnostic than you can get by these physical biopsies. This is Hannah Valentine, our collaborator right here, who I mentioned earlier. This is Tom Snyder, the postdoc in my lab who did the work. So We did this on a small number of sort of bank samples and published it in 2011. We're very excited about it. It was just about the time of when we were doing this work that the Obama stimulus was going through. That gave a lot of money to the NIH, and they were putting out calls for proposals for things like what we were doing. So we actually were fortunate enough to receive a rather large grant, which let us do a large prospective trial of this in the Stanford Hospital, involving a large number of physicians and patients. Close to 100 patients agreed to be part of the study. Most of them from heart, some from lung, and we got from them a total of 700 blood samples. And in the heart, we were able to show that it's actually a pretty good diagnostic tool. This is a receiver operator curve, area under the curve of 0.81. Which is far better than anything else that's out there right now. And we also, with the lung patients, were able to start doing proof of principle and get the sort of first examples that it does seem to correlate with rejections, both here and here. The patients were treated for rejection. And so that for us is very exciting, and we're looking forward to seeing the next steps that will bring this sort of diagnostic into the clinic. So I'm going to change gears right now and take you in another direction. I've talked a little bit about the immune system and what happens when it starts to attack foreign things in your body, like a foreign heart, someone else's heart. But of course, the immune system protects you from pathogens and all sorts of other assaults on your body. And I want to tell you a story about how we've been trying to use these sequencing technologies to create kind of a unique measurement of the state of your immune system, with the goal of getting some sense about what is a healthy immune system. If you go to your doctor right now, you can't ask your doctor, how good is my immune system? Strong, weak? They just don't really have a measurement for that. They're using things that were developed back in the 40s, and they're very primitive ways of looking at your blood cells. And so let me tell you a bit of a story about how antibodies get made. So you've got two parts of your immune system, your innate immune system, your adaptive immune system. I'm going to talk about the adaptive immune system, which is the part that creates antibodies and molecules which are molecules that can recognize virtually any foreign thing that comes in your body. It's pretty spectacular. This is why you get vaccinated, to train your immune system to make antibodies against certain diseases. So this is a picture of an antibody. The structure, it's a protein molecule, and like I say, it recognizes foreign molecules that come in. The business end is circled in green. There's two of them. They're sort of symmetric. And the cells that make antibodies are called B cells. And there's sort of a really neat combinatoric process that happens to make these antibodies. Because a huge amount, they're sort of little models of evolution, a huge amount of diversity is generated. And then the ones that work are selected out by the immune system. So one way in which diversity is created is by having a bunch of little gene fragments that get glued together. These are like little Lego blocks. And there's basically 3 different flavors, which are shown here in different colors. They've got names, the V's, the D's, and the J's. And what each B cell will do is it'll pick one of each flavor and then glue them together to make a new antibody gene. And so the commitment of these B cells to making antibodies is so strong that they reprogram their genomes to make one and only one antibody. The picture in your head to have is Cortez burning his boats when he arrives at the New World, right? There's no going back once a B cell decides to make an antibody. And so they all do this more or less randomly, and that creates a certain amount of diversity. You can multiply out the numbers, and it's something like 10,000 different possibilities at the end. That's not a huge number as far as one's desire to recognize different things goes, and so there's other ways that these cells will create diversity in the antibody genes. One is that they're not very careful about how they glue these 3 things together. They don't do it very cleanly, and in fact they can nibble away at the sequences and add random nucleotides there, and that creates what's called junctional diversity. So by creating random sequences at the 2 junctions where they go together, creates another amount of diversity. And then there's a process called somatic hypermutation, which is to say this B cell can go and create random point mutations anywhere in the antibody gene. And that creates another layer of diversity. And if you work out all the numbers, you can do some back-of-the-envelope estimates about what's the total possible combinatorial space of antibodies. Let's look at human right here. These are the V's, the D's, the J's. You multiply them together, you get about 10,000. And then you look at these other sources that create a heavy chain diversity, about 10 to the 7th. Then there's another thing called the light chain. Don't worry about that. But more or less the total number of people carry around is around 10 to the 14th possible antibodies that could be made by these mechanisms. That's a huge number. That's a lot. That's, we're talking astronomically large. In fact, it's too large to fit into one person. The total number of B cells you have in your body is about 10 to the 10th, and you can't have more antibodies than you have B cells, because remember, each B cell commits to making one and only one kind of antibody by reprogramming its genome. So that's the upper practical limit. This is the theoretical limit. And as I got into this field, kind of wondered as a naive physicist, how many antibodies are there in a person or in any organism? And it turns out that wasn't known. All right, so the back of the envelope calculations were sort of there. There were various estimates in the literature that were all over the map, and nobody really had the measurement tools to answer the sort of really fundamental question of what is the frequency and distribution of antibodies in a given individual. And this is something we decided to attack with these sequencers. And in this case, it was a very talented student named Josh Weinstein, a postdoc named Jenny Jiang, and our collaborator Daniel Fisher. And we decided to humans, 10 to the 10th, still a big number, plus there are practical difficulties in sort of exsanguinating your donors if you wanted to get all the blood. So we decided to look for a model system. And we settled on fish, in particular zebrafish. It turns out that the adaptive immune system, this ability to make antibodies, first came into being about 400 million years ago when fish got jaws. So somewhere in the evolution of vertebrates, when fish got jaws, they began eating things that required them to have a more sophisticated sense of self and foreignness. And so if you look back, shark, they have a primitive immune system that makes antibodies, zebrafish, coelacanth. And it turns out that zebrafish, besides being a monogenetic organism, also have all the same sort of biochemical mechanistic methods of creating diversity that people do, and which I just described for you. They have this ability to do the combinatorial rearrangement of the V's and VD's and J's. They can do this junctional diversity by nibbling at those junctions, and they can do somatic hypermutation. And so that seemed to us like a really good model system. And it wasn't something that had really been done before, but we decided to explore that. Plus, they only have 10 to the 5th B cells, so something reasonable to measure. So our experiments went something like this. We take the fish and we would grind them up and make RNA out of them. So we don't care about their physiology and where the B cells were hiding. And well, if you ever, some older people in the audience, right? The old Saturday Night Live sketch, The Bassomatic. I sometimes show it in the talk. But it turns out that truth is stranger than fiction in the end. I stumbled across this article from the BBC that liquidizing goldfish is not a crime. It turns out that some artists had created an exhibit where they turned a blender into a fish tank and plugged it in and kind of dared the patrons of the museum to push the button to turn it on as they went through and looked at it. And they did. That tells you something about human nature. So the police hauled off the curator of the museum and arrested him and charged him with cruelty to animals. And at his trial, the blender's manufacturer, Moulinex, had an expert witness who testified that the fish had died painlessly within 1 second. And so the court let him off. He didn't have to pay his fine. I've been using this slide for a while, and one of my colleagues who works on fish genetics saw it. He asked me for a copy of the article because he needs something to show the animal protocol people that the way he treats his fish is humane. Do that. All right. So that's more or less what we did. We were able to amplify out the RNA from the expressed antibodies. For the experts in the audience who are interested in the heavy chains, make a library, sequence it, and read out the interesting sort of business end of the molecule for tens of thousands of these molecules per fish. And so it gives you an enormous amount of sequence data, which you can do you can basically compute on it, and it starts to look like a physics problem because you consider all the statistics of these sequences and how they relate to each other and what the correlations are and any structure that might be there. And it's a really fun problem. This is what the immune system of a fish looks like in sort of a coarse view. Remember these VDJs. So this is how the VDJs are used. These are 4 different fish. This is the V-axis, the D-axis, and the J-axis. So every point in 3 space represents one unique VDJ combination. And the size of the ball and the color tells you how many antibodies use that VDJ combination in that particular fish. And you can see that they all look really quite different. And so that's unfortunate. These fish are all different. What's the common denominator? Is biology going to yield anything universal out of it? Well, it turns out it does. So if you just look at the frequency distributions now for many of these different fish, they do all fall on sort of a universal curve. This is a sideways histogram of fractional abundance and number of VDJ combinations without abundance. And what it says basically is that there tends to be one VDJ combination that dominates the abundance and has the highest abundance. And there's a lot of VDJ combinations that are used very infrequently. And that relationship is very similar in all the organisms, all the fish, all the individuals. You can also do this down at the level of antibody sequence. So get out from this coarse grain view, zoom in, and look very closely right down the level of mutations separating sequences. And again, you see sort of a universal curve and something that physicists love. It looks like a straight line on a log-log plot. So we did a lot of things with this data. One of the things was to estimate the diversity. That was our first question. And it turns out there's 3,000 to 4,000 different antibodies in a given individual fish. And a lot of other cool things came out. I'm not going to dwell on it now. We've also taken what we've learned with this model organism and moved on to doing studies in humans, some support from the Simons Foundation, in fact. I'll leave you with this sort of last thought about what measurement technology brings to problems like this. This is the total number of antibody sequences measured in the history of the universe. Okay? And it started for all practical purposes in the early 1990s. And by 2008, there were maybe 80,000 antibodies that have been sequenced. Our one paper on those zebrafish changed the curve dramatically. This is now what happened one publication later. Enormous increase in the amount of data we've got about these molecules. And I think there's a lot to extract from that. It's particularly great news if you're a zebrafish, but of course we're now doing these measurements in mice and humans as well. And it's leading to hopefully what will be quite a different and powerful way of looking at the immune system. So let me change gears again. Go back to another physics metaphor. So dark matter and physics. You've probably all heard about that. People, by making these sort of indirect observations, either sort of rotational behavior of galaxies with respect to the known mass, or by looking at lensing effects of the Space Telescope, we're able to infer that there's a lot of mass and energy out there that we can't observe directly. And this is the bit that you see directly right there. The rest of it's all dark, and that's not a great situation to be in. Unfortunately, it's not my problem to have to solve. It's someone else's problem. But it turns out biology has a similar sort of challenge. And there is a problem of dark matter in biology, which is to say that the vast majority of biological diversity on Earth is not observed directly, and it's only inferred indirectly through measurements that are not quite the same as I showed you about angular momentum and gravitational lensing, but through other means. And I'll describe that for you in a little more detail now. So let me tell you how life is classified. This is the tree of life right here. The tree of life is constructed by looking at the evolutionary relationship of a particular gene. There's a number of genes that are universal to all living things, and one was chosen to make these trees sort of for technical reasons because it was easy to measure. It's sort of a historical artifact, but it's what we've got. This is what you get when you ask, how does this change from one species to another? Species that are very close to each other, just one or two mutations away in this gene, will be close on the tree. Ones that are many mutations away will be farther apart. And you can sort of work out a structure of relationships in that way. And in case you had any illusions of your special place in the universe, all right, you are, as a human, in this animal branch. The closest things to you are fungi to the right and slime moles to the left. All right, so think of how proud you are to be different than a slime mold and a fungus. Okay, and that's just this little bit of the evolution of the tree of life. And this is all the rest of diversity that's out there. I mean, it's really phenomenal. And the vast majority of it are single-celled microbes, bacteria and archaea, which are the blue and the red here. Okay. Of those microbes, only about 1% can be grown up in laboratory culture. I mean, that's the way microbiology works. You take a little microorganism, try and grow it in a test tube or a petri dish. If you can grow it up, you can study it. You can do sort of biochemistry, microbiology, wherever you want on it. Can't grow it up, you're kind of stuck. And so for 99% of what's out there, folks are stuck. That's the dark matter problem. And here's another way to kind of get your head around that. If you think about the sort of Linnaean classification system, how things are given, the nomenclature by which they're given their names, kingdom, phylum, class, order. King Philip came over from Great Spain, that whole thing, genus and species being the most finest order, kingdom being the grossest way to separate animals, separate organisms into different kingdoms. After kingdom comes phylum. So the second grossest way to separate things is shown here, okay? And so this is for a bunch of this is for the microbial, this is the bacterial kingdom. And all the ones that are in gray are different phyla for which there's no cultured member. All right? It was dark matter, and its existence is only inferred by the analysis of that 16S sequence, that one gene that was floating around. And so there's a lot of them. Even at the phylum level, nothing is known except that something exists there. And I'll draw your attention to this one right here, TM7, which is right in the middle of a gray desert. And this was one of the first ones we focused on to try to get some information about. And the way we did that was through these microfluidic tools that I mentioned earlier. So these are basically small plumbing, ways to create chips that have, this one had, 600 or 700 valves on it. We're now making ones with tens to hundreds of thousands of valves, and use them to do biological experiments, to do fluid automation and manipulation. And this chip is a picture of an actual chip. The channels have been filled in with food coloring, and we took a picture of it through a microscope. The channels themselves are on the order of a human's hair in width, 100 microns or so. And we spent sort of the last decade kind of developing this technology and using it to look at a variety of different biological problems. One of which is how to sequence genomes from single cells without having to culture them. And in a series of experiments with a variety of collaborators from the microbiology community, we've been able to generate single-cell genomes from a variety of interesting organisms, some of which were meant to help fill out this tree of life. So our first one, the TM7 I mentioned right there in 2007. Also, we've done things at OP11, OP9. These are all sort of gray things on that tree. We've done things that are interesting from the sort of environmental point of view. So turns out that this particular one, which is an archaea, is involved in oxidizing ammonia, and lives in the San Francisco Bay and many other sort of saline environments around the world. It's involved, when it gets out of balance, you get things like red tides. So it's a very important component of keeping the balance of ammonia in seawater from getting to be too great and feeding those red tides. This is one that is thought to be involved in, this is a mouse version of a bug that's thought to be involved in various human diseases, including cancer. This was one we did for fun through a class. This is the fastest swimming bacterium that's known. It goes like 100 microns per second. It's a very good swimmer, and it couldn't be grown in culture. You could watch it swimming across the microscope slide, but it couldn't make it reproduce for you. Fortunately, we could catch it, and we're able to amplify the genomes and sequence it. And a picture of the chip that we use, in which we basically were able to use the valves to isolate single cells in a chamber, and then move them from one chamber to the next to do a series of steps to break them open and amplify their genomes and recover the amplified DNA from the chip for sequencing. So we were having a lot of fun doing that, and we realized that thanks to this wonderful Moore's law, the Moore's law equivalent of sequencing getting cheaper all the time, that it was actually becoming practical to do single-cell genome sequencing from humans. And so, with a few modifications from the chip, we decided to do that. And there's a bunch of reasons for that. One of which is this thing I referred to earlier, or alluded to earlier, that not all cells in your body have the same genome. And there's a variety of examples of that. One that we've been talking about is the immune system. Remember, these B cells that make the antibodies, they've all reprogrammed that one gene in their genome, so it's all different in all of them. Another example are your gametes, sperm and egg cells, right? So those are haploid, they're not diploid, because when you want to make a baby, you want to have it half the genome from the mom, half from the dad, and the way that happens is by making these cells that are haploid. They combine and create a diploid child. And so that's one way in that they're different. Another way in which they're different is that they mix together the two halves of your genome in a way that I will describe more fully in a couple of slides. But the other broad class of cells that sort of have genomes that vary within your body are the ones that are going to give you cancer, which is to say, every time as you age, every time one of your cell divides, it makes a mistake when it copies the genome. Many of those mistakes are innocuous, aren't going to cause you trouble. Occasionally, those mistakes will happen in the middle of important genes. And when that happens in enough genes, the cells can change their character quite a bit. Instead of being well behaved, they'll grow without restraint and create tumors. That's essentially how cancer happens. So you can think about cancer as an end state of aging on the one hand, but as a case where there's an enormous amount of genomic heterogeneity, both within the tumor and between the tumor and the rest of the body. All right, so if there's time at the end, I may tell you a bit about that, but let me tell you a story about gametes and sort of symmetry, which all physicists love. So, while I was doing my literature search before starting this project, went back to figure out what the ancient Greeks knew about sex back in antiquity, and they had very sophisticated theories of development actually. So they believed that everything followed a mathematical law. They had theories of how human development followed mathematical laws. They had amazing knowledge of how a human fetus develops inside the womb. I don't even want to imagine how they got that knowledge, but they had it. They tried to make it fit all kinds of mathematical formulas that were quite interesting. And they had all kinds of other theories about human biology. So Anaxagoras thought correctly that male determines the sex of the baby. That's correct, right? Women are XX on their sex chromosomes, men are XY. The sperm contributes an X or a Y, that determines the sex of the baby. He was right about that. But he thought that the male seed, or the Y chromosome, came from the right testicle and the female seed, or the X chromosome, came from the left. We know that's not the case today. But that did not stop early bioengineers from trying to exploit that theory and control sex of offspring by using ligatures to tie off one testicle or another, which they apparently did on bulls. There's references about that, but I've not seen the final paper that they published on that. And there's more generally these ideas that left and right were quite different, and there's a strong amount of asymmetry in biology. Milk from the right breast was stronger and more suited to male infants. Diseases of the right side are more severe. And even Aristotle had general theory, biological theory of left and right in terms of where the organs were and development happened. As that leads to sort of a natural question, how much of this was bunkum and how much of it was right? And in particular, is there a left-right asymmetry in the male gonads? And it turns out to be an anatomical fact that the right testicle is, for the most part, positioned higher and is heavier than the left one. And this was validated by a crack team of pathologists in 1960 who apparently had nothing better to do with their time than to cut up male cadavers and weigh their testicles. And they're able to show that there is this very general asymmetry. And it also turns out to be true that the Greeks knew about this. And how do we know the Greeks knew about this? Well, this very diligent graduate student in the 1970s, Mr. McManus, went around the British Museum and looked at all the statues that had survived from antiquity, and he measured the size and position of the testicles. And he was able to show, as a function of the age of the statue, that the percent of statues whose testicles are the same size and height was decreasing over time. Pretty amazing. And there's our friend Anaxagoras right there, right in the middle of that. So he maybe was onto something there. This was a very silly year for Nature magazine. There was a whole series of papers that came out after this about this phenomenon. It was endless. And this work was so influential it won Dr. McManus the 2002 Ig Nobel Prize. Now, jumping forward to modern times, we are quite interested in symmetry and asymmetry in a lot of different fields, but now, not so much in the physiological asymmetries, but in the molecular symmetries and asymmetries in these cells. And, in particular, how genomes get partitioned up in the germ cells, in the sperm and the egg. And so I told you I'd say a bit more about that. Let me give you a slide that kind of illustrates that. So you get half your genome from your mom, half your genome from your dad. Which one do you give to your kids when you have children? Neither, both. You give a mixture. right? That's what meiosis does. So remember from high school biology, mitosis is how cells divide when they just want to grow and create new organs. Meiosis is the special division process that happens in your germ cells. And what it does is it creates a whole series of unique genomes by mixing together your paternal and maternal inheritance. And that's sort of described here in this cartoon. So imagine you had a diploid organism that had only 2 chromosomes instead of the 23 that we do. And so here's the blue ones, this cell inherited from its father, and the red ones from its mother. And now it undergoes meiosis. So these chromosomes duplicate and associate with each other. And when they touch each other, there's the machine that can, a biochemical machine that can cause them to swap parts over. So the homologous chromosomes come together, and there can be some mixing of them to create these sort of mosaics. And then the cells divide and divide again to create haploid cells. And now, instead of putting random sort of pure maternal or paternal inherited chromosomes in them, it puts these mosaics in. Okay? And so each of these germ cells, the sperms and the eggs, have unique genomes that have never been seen before and are essentially random mixtures of your paternal and maternal inheritance. Okay? That's called recombination, and that's one of the really important ways in which sperm and egg cells differ from all the rest of the cells in the body. And this recombination process is a very important contributor to genetic diversity among humans, and there have been substantial large-scale efforts to map that diversity across the entire human race through projects like HapMap and Decode. And we got interested in trying to measure this recombination diversity at the level of one person. We wanted to ask the question, how much genetic diversity gets created within one person's germ cells, and how does that compare to the diversity that's been observed across the human race? And so we decided to work on the ejaculome. So the experiments are pretty straightforward. It starts with sample collection. No microfluidics, nothing small about this here, very low tech. And then the sperm is put into the chip and separated out. So the cells are randomly dispersed in different chambers. They're lysed, the genomes are amplified, and the amplified genomes recovered from the chip and then analyzed by genotyping, by sequencing, and a few other methods. So from each cell, we then take that genetic information and map it back to the sperm donor's genome, okay, which we've not only sequenced but haplotyped. So we know for this person which part of the genome came from their father, which part came from their mother. We take each sperm cell, map the information back, and see which of the 2 chromosomes it goes to for any given one. And where it crosses over from one to another, we say, aha, there must have been a recombination event there, and this particular sperm cell has this unique mosaic chromosome. Right? And we do that for all the chromosomes and all the cells we measured, give us a total of about 2,000 recombination events, which is a very large data set in this business. And here's an example. So for every cell, we created a synthetic map like this, which shows for each chromosome what the recombination events were. So for this particular cell, chromosome 1 was largely from the paternally inherited part, but this crossover event put in a little bit of the maternally inherited part right there. 2 is a little more complicated. The top part's paternal, top part's maternal, middle part's paternal, lower part is mostly maternal, except for the very tip down there, and so forth and so on. And so we have these maps for every cell. We can then compare it to the population-wide data, which is shown here. The lines are the 2 population-wide measurements that were done by HapMap and Decode, respectively. And the circles are our data. And if you start doing statistical comparisons, it turns out that they're more or less indistinguishable. And you see just as much recombination diversity within the germ cells of one individual as you do across the human race, which is kind of spectacular. And I call that an ergodic principle of recombination. We took a few of these cells and sequenced them very, very deeply, so we had the whole genome out of them, and found that in some parts of the genome, as we compared the data from the single sperm cell back to the donor's genome, there was just complete disagreement with the donor's genome for a very small number of sites. And we eventually realized that those were de novo mutations, new mutations that were unique to that sperm cell that weren't part of the somatic genome of the donor. So we measured the rate at which that occurred It turns out the rate for this individual is about 3 times 10 to the minus 8. Without getting into too many of the technical detail, it turns out we were right. And later experiments that measured that property by a different method came out with numbers that are completely consistent with ours. We're quite happy about that. And it's sort of a useful thing to know. So in the last couple of minutes, let me turn to the example of cancer, which, another case where there's an enormous amount of genomic diversity from cell to cell. And I can stop now if it's getting too late. That's okay? All right. It's a sort of one of the very powerful examples to look at and to use these tools. And so this is work we did in collaboration with Ravi Majeti, who's a young physician at Stanford, his student Max Jan, and my postdoc Tom Snyder. It's sort of a complicated experiment, but let me sketch it out. These patients had leukemia, so cancer of the blood. We sequenced their tumor, all the cancerous cells in their blood, and we also sequenced their somatic genome. And we looked at the differences between the tumor genome and their normal genome. And we found a number of mutations. Many look quite interesting. And we took some of those interesting mutations, and then we went back and we looked in their blood. There's most of the blood is tumor, but a little bit had not been turned into cancer. So there's residual, what are called normal hematopoietic stem cells left in the blood. And we said, do these normal cells have any of the mutations that were part of the tumor, but not the rest of the normal genome? And it turned out there were. A very small fraction, but some of these cells had mutations that were part of the tumors. These must have been the earliest mutations that caused the disease, the very first few that happened. And we got interested in trying to understand, how did those mutations come about? What was the order in which they occurred? And so we started to do single cell measurements where we could look at many of these mutations in many individual cells. And I'll give you a picture of that on this next slide. For this particular patient, each axis is a set of is a mutation or pair of mutations that always occur together. And each blob is a different single cell that we measured. And you can see that for most of the cells, almost 500 of them, none of those mutations were there, mostly normal. 50 of the cells had this mutation. 3 of the cells had this one and these two. And 1 of the cells had all of them. And you can see that it was never the case that this mutation occurred without these two and this one. And it was never the case that these two occurred without this one. So it's a perfect Venn diagram in the way these mutations occurred. And so you think about that for a minute, you realize you can write down what was the natural history of this tumor, of this cancer. And we know that this mutation must have come first, these were second, this was third, and somewhere later it acquired another mutation that caused these cells to stop being normal and turn into a tumor, and it grew into leukemia. And so this ability to sort of map out the natural history of a naturally occurring tumor is something that we're diving into very deeply now. We think it's going to be an important tool in understanding the origins of cancer and perhaps in understanding how to better treat them. So I think I'll wrap up here. I've tried to acknowledge the folks along the way who've done the work. I told you at the beginning that these were the 2 measurement technologies that were having a sort of profound influence over my work and many other people in the field, both the sequencing and the microfluidics. I gave you examples in areas ranging from prenatal diagnostics to organ transplants to immunology and finally to human reproduction and cancer. And just in case it all sounds so irresistibly fantastic, let me leave you with a few words of caution from Maurice Wilkins, who was one of the Nobel laureates honored for his work and his role in discovering the structure of DNA. Thank you.
Thank you. We have time for some questions. Back here.
Do you know what the gene, that initial mutation SKP2, what that encodes?
SKP2, yeah, that's a well-known oncogene, actually. That's one that's known. So many of the genes that were popping up are ones that have been known to be associated with cancer and to be important, but the order in which they occurred was not known. Yeah.
Could you use the looking at the DNA fragments in blood to diagnose cancer, detect it?
That's not something we've been working on, but others have. There's a literature stretching back decades about that. And it's potentially quite useful. Somehow it's never made it in the clinic, despite many beautiful papers, but I think its time may be coming now.
Is it true that biology experiments are not as easily repeatable as, say, physics experiments?
Yeah, so that's the complaint that physicists always make. You know, it's true that in physics, you know, you don't have to keep everything in the fridge and have it go off. But, you know, how to say it? I'm trying to think of a counterexample in physics, you know, that is not repeatable. But I would say you pick the right problems, the right systems, everything's repeatable. Let's say that in biology.
High temperature superconductivity.
Yeah, there you go. That's a good one, actually. All these materials, one-off things.
Can you tell the difference between a sick zebrafish and a healthy one from looking at the VDJ frequencies?
From the immune system. You know, that's a really interesting question. We're trying to answer it right now. Not with zebrafish, but with stickleback fish, which are, you know, for technical reasons, we decided to try to look at them. And the data is rolling off the sequencer even as we speak. I'm quite curious to see what it's going to be.
You seem to imply that invertebrates' immune systems were different.
Mm-hmm.
Fish. Can you say anything simple about that?
Oh, yeah. So, you know, many creatures all the way down the evolutionary time scale or ladder, everyone I think about, have immune systems. They have innate immune systems that are kind of pre-programmed to respond to different challenges. Even bacteria, it turns out, can have an immune system they use to fight off viruses and its phages. And that's getting a lot of attention right now because the details have been worked out. But as far as this ability to create antibodies and molecules that recognize, that really happened first in jawed fish. And people in evolutionary biology refer to it as the Big Bang because there was a lot of duplication in the genome at that point. And that's how all those gene fragments came about. There's a lot of gene duplications. And so you can see it's correlated with a sort of increase in the size of the genome and the amount of repetition that was there.
You said that 500,000 women undergo this non-invasive amniocentesis.
Yeah.
How many undergo actual amniocentesis?
Oh, God. I mean, it's a huge number. You know, I think there's, 60 million pregnancies in this country every year, something like that. And, you know, a pretty good fraction are getting amnio. I mean, it's been, you know, sort of historically, they recommend only women over 35 and higher risk. And now they're kind of rolling that back and trying to say that, you know, pretty much all women should get it. So the OBs are getting pretty confident in their recommendations.
Can a woman choose to have the non-invasive if she chooses, or is it only available in certain places?
So that's a good question. It's available in the US. It's available in China. There's a few countries in Europe where it's starting to be available, but it's certainly not available worldwide right now.
If you look at the same leukemia, ostensibly the same leukemia in different patients, do they have the same sequence of progression from the same type of mutation one to the next?
That's a really good question. So we did look at the same leukemias, AML, and acute myeloid leukemia, in about half a dozen different patients. It was always different genes. It's a very idiosyncratic disease.
Could you repeat the question?
Oh, she was asking if we look at the sort of the same cancer type in different patients, do you see the same genes popping up in each of them? And the response is no.
I have a question. You described the recombination as essentially being a random mixing.
Yeah.
One thing about the pictures you showed was clearly not random, and that was there were very few crossovers.
Between one chromosome and another, you mean?
Yeah, look down one chromosome and had 4 or 5, 3 crossovers rather than several hundred thousand.
Oh yeah, all right. So that's, all right, I see. Just the number that are occurring. So the average is 1 per chromosome, and there's fluctuations around that. And, you know, it's interesting that, you know, it's, there's enough apparatuses that you pretty much always get 1 per chromosome. So it's not purely Poisson distributed. You don't get as many zeros as you'd think, given the numbers. But, you one thing I should also say is that the sort of this universality that I was talking about, as you compare a low resolution across human diversity, it is known that individuals, that uniformity goes away as you look at finer and finer resolution. So there's so-called hotspots that are recombined much more frequently than others in the genome. Those have been measured and characterized at the population level and to a certain extent individual level. And we see that behavior too as we drill down to finer and finer resolution in those studies. And there's interesting things about the single recombination map that.
You mentioned the chromosomes bumping into each other. Some mechanical explanation for what John's talking about?
Well, there's no action at a distance, let's put it that way. So the machine that does the recombination requires, he's got to grab both the arms and put them together and cut and cross. And so it's a very physical process.
And there are weak places or strong places in which it will happen.
Well, this is where those hotspots come into play. So there's certain favored places, and some of them are associated with protein binding sites. So it seems like, a half to 2/3 can be explained by, a particular DNA sequence is recognized by a protein called PRDM9, and that drives part of it, but that's not the whole story. Yeah.
You mentioned that recombination basically is not, the probability of recombination is not uniform along the chromosome. Is there any correlation you can see? So let's say if you see the recombination particular point, either correlation with a similar event in the other point.
Yeah, so the question is, do we see correlations in recombination events? There's a very complicated story around that called gene conversion, where you can sometimes see that just a small piece is moved over, so you're having two correlated recombination events right near each other. That's been studied a bit in the literature, and we see some evidence for that, although our signal to noise is not as good as I'd like it to be in that particular question.
The non-invasive procedure for Down syndrome, how soon can it be done? And that's one. And as the accuracy of your test increases, if you do it later.
Yeah. So the question is about the Down syndrome test and when can you do it and how does the accuracy depend on the gestational age. Right now the commercial guys, I don't know, somewhere in the 10th week, I think they're doing it. I personally think you can do it almost as early as you like. Often the first doctor's visit is somewhere around the 8th week, and there's enough cell-free DNA there that I think you could do it and get a good measurement. And because of the way it works, this counting principle, the higher you count and the more fetal fraction there is, the better it works. So the later in pregnancy you do it, for a given number of counts, the better your answers are going to be. And the fetal fraction does go up through the course of pregnancy. So, but the whole charm of the counting principle is that you can make it as good as you want, your sensitivity and specificity, simply by counting higher. So if you wanted to do it very early in pregnancy, where there was a smaller fraction of fetal DNA, you could make it be as good as you want, simply by sequencing deeper and getting more counts.
If I'm interpreting what you said about the diversity and this stuff, it seems like a pair of parents can only have a small number of relatively small number of possible kids. Is that right?
Well, define small.
It's smaller than 2 to the number of bases, okay? Because you're saying that there's...
I got an astrophysicist here. Astrophysicist, right? Yes, much smaller than 2 to the number of bases, absolutely. What's that?
How big or how small?
Yeah, so the number to keep in mind is, there are, pick a number, let's say order of 50 de novo mutations in each child. So you get 50, you can, 3 times 10 to the 9th, choose 50 or something like that for your de novo mutations. And with the recombination events, there's on average 1 per chromosome, so something like 24 per sperm and per egg, and you can work out the number of places that can happen. And I won't do it in my head, but you can go do it in the wine break. Those are the numbers you put in the calculation.
They're too big to have any real meaning.
Today, how does the cost, just the financial cost, of doing the new procedure for looking for Down syndrome compare with the amniocentesis?
All right. The question is about the cost of the Down syndrome test. It's interesting. When we published our paper, everyone said, "Oh my God, the sequencing costs are so expensive. It's going to be, just a boondoggle and ridiculously expensive." Well, it turns out that funny thing about that exponential drop in sequencing costs. Sequencing are not the most expensive part of the test anymore by a long shot. The way most people are pricing it, it is below the cost of an amniocentesis. In this country, amnio costs around $3,000, and these tests are priced around $1,500 to $1,000, and so cheaper than the existing procedures.
Are they covered by insurance?
Some of them are starting to be covered, yeah, and that's what all the companies are working very hard to do, is to get them all covered.
Limitation of this test. So, for example, if it's multiple pregnancies, like twins or triples, so you cannot tell which one.
Yeah, that's right. So multiple pregnancies are excluded right now, but there are interesting ways to try to measure that are fun to think about.
All right. Let's thank Stephen Quake once again.